right this very second, as you sit there listening to this, a quarter of all the energy your body is burning is being funneled into, well, one microscopic, totally invisible task.
Right. Yeah, it's pretty wild.
Right. Like
Yeah.
you aren't using that energy to think or to breathe or, move your muscles. You're using it to operate billions of these tiny molecular turnstiles on the surface of your cells.
Exactly. And I'm glad to be here to unpack this with you.
Yeah, it's great to have you. We are doing a deep dive today into something you, the listener, might have just dismissed as a boring biological wrapper. I mean, the cell membrane.
Yeah, people tend to overlook it.
Totally. But we have a massive stack of historical research and anatomy notes to get through today because, well, we are looking at the ultimate gatekeeper of your body.
We really are. And, if we look at the visual backdrop we've set up for this deep dive, it's this dynamic 3D rendering of that bustling cellular surface.
It looks crazy, like a crowded microscopic city street or something.
It really does look chaotic at first glance, but fundamentally the cell membrane, or plasma membrane, is what we call a selectively permeable phospholipid bilayer.
Right, selectively permeable.
Right, meaning it chooses what gets in and out.
Yeah.
And it's studded with proteins and carbohydrates and cholesterol. It completely regulates what substances are allowed to cross into and out of the interior of the cell.
Okay, let's unpack this, because before we get to the massive energy toll of those molecular turnstiles you mentioned, we really have to look at the wall itself. Like, how does a microscopic border wall actually get built?
It's a great question, because when you look at the building blocks, these molecules called phospholipids, they have this intense split personality.
Okay, split personality how?
Well, they are what we call amphipathic molecules.
Amphipathic.
Right. Each individual molecule has a polar phosphate head, which is hydrophilic, meaning it loves water.
Okay, got it. Heads love water.
Yep. But attached to that head are two nonpolar fatty acid tails, and those are hydrophobic. They completely fear water.
So if you drop them into the watery environment of the human body, they face this like massive physical problem.
Oh, totally. The heads want to be in the water, but the tails are absolutely terrified of it.
It's kind of like a massive crowd of people caught in a sudden downpour, right? Like where everyone's feet absolutely hate the rain.
That's actually a perfect way to picture it, yeah.
People are going to automatically huddle back to back, so the heads face outward toward the rain, and the tails hide on the inside, completely shielded from the wet environment.
Exactly. And that huddling is what forms the bilayer. Two stacked sheets.
But wait, if I'm picturing this right, there isn't some cellular construction crew putting this together, is there?
No, not at all.
Like there's no DNA blueprint actively directing a protein to lay these bricks into a wall?
None. What's fascinating here is that the self-assembly is driven entirely by thermodynamics.
Just blind physics.
Just physics. Specifically, the hydrophobic effect. The cell doesn't use a single drop of ATP, or, energy, to construct this foundational bilayer.
That is insane to me.
It is. If you have amphipathic lipids and you have water, the basic architecture of a cell membrane is chemically inevitable.
Inevitable.
Yeah, molecules spontaneously assemble into that back-to-back sheet simply because it's the most energetically stable arrangement possible.
So the most critical boundary for life just forms itself just because of how the chemicals react to water.
Pretty much.
But this brings up like a logistical nightmare for early scientists, right? Because this boundary is just nanometers thick.
Oh, yeah, incredibly thin.
So if it's completely invisible to the microscopes of the late 19th century, how do you even begin to prove what it's not of?
Well, you have to stop trying to look at it, and you start testing its physical properties.
Okay, how do you do that?
So in the 1890s, a botanist and pharmacologist named Charles Ernest Overton, he started measuring how quickly hundreds of different chemical compounds entered plant cells.
So Overton is measuring what gets in. But how does that help him if he can't see the wall? I mean, if I throw a tennis ball against a wall in the dark and it bounces back, I know the wall is there, sure, but I don't know what it's made of.
Right, but you would if only certain types of balls could pass through it.
Oh, okay.
Right. Overton noticed a highly specific pattern. Substances that dissolved well in lipids, like oils and fats, crossed the cell boundary far, far faster than water-soluble substances of a similar size.
Wow, okay.
So from that purely functional data, he deduced that the invisible wall must be made of lipid. He mapped the composition of a structure he couldn't even see.
Which sets the stage for the 1920s, right? When two Dutch scientists, Evert Gorder and Francois Grandel, they finally figure out it's a bilayer.
Yes, and they do this by extracting lipids from red blood cells.
Because mature red blood cells don't have a nucleus or internal organelles, right? They're basically just pure membrane.
Exactly. So they take these extracted lipids, spread them across a water trough, and they just measure the surface area.
And what did they find?
Well, they realized the lipid film covered exactly twice the surface area of the red blood cells they started with. 2 times the area means 2 layers.
Because it was a bilayer.
Right.
They essentially unfolded a 2-story house into a single flat driveway. But, and this is wild, when historians go back and look at the actual math of their experiment, the numbers shouldn't have worked.
No, they absolutely shouldn't have. They made 2 massive errors.
Yeah, they underestimated the surface area of a red blood cell, so they thought the cell was smaller than it was. And on top of that, their extraction technique was highly flawed, so they didn't even get all the lipids out.
It's honestly hilarious.
It's like trying to calculate the paint for a house, measuring the house as half its actual size, spilling half your paint on the lawn, and somehow magically having the exact right amount to paint the house perfectly.
That's exactly it. The errors canceled each other out almost perfectly. An incredible stroke of luck for science.
Unbelievable.
But, establishing the bilayer was really only half the battle. By 1935, Hugh Davison and James Danieli proposed what they called the protein-lipid-protein sandwich.
The sandwich model.
Right. They pictured the lipid bilayer sandwiched tightly between 2 continuous rigid sheets of protein.
So kind of like a cracker with a soft lipid filling.
Yeah, a rigid cracker. And this static model was incredibly dominant. It held sway in textbooks for about 35 years.
Okay, wait. So what does this all mean for how science works? Because if everyone believes the membrane is a rigid cracker sandwich, how does a cell actually change shape or divide? A rigid cracker snaps under pressure.
That physical limitation is exactly what started bothering researchers, and it was compounded by new imaging technology in the late 1960s.
Better microscopes.
Exactly. Freeze-fracture electron microscopy. It allowed scientists to literally split a frozen membrane right down the middle of the bilayer.
Oh, wow. And what did they see inside?
Well, when they looked inside the lipid core, they didn't see a smooth filling. They saw these globular proteins embedded deep within it.
So the proteins weren't just sitting on top like crackers.
No, they plunged right through the lipids.
And then, the 1970 Fry Edison experiment pretty much proved the whole thing was actually fluid, didn't it?
It did, yeah. It was a brilliant experiment.
They took a human cell and tagged its surface proteins with a red fluorescent marker, right? And then they took a mouse cell and tagged its proteins with a green marker.
Yep, and then they fused the 2 cells together.
So if the static sandwich model was right, that fused cell should just look like a 2-tone car, half red, half green.
Exactly. But within 40 minutes at body temperature, the red and green tags intermixed uniformly across the entire surface of the fused cell.
They completely blended.
Yes, it was direct, undeniable proof that membrane proteins move. They drift.
That means right now, on every cell in your body, the proteins aren't sitting still. They are actively drifting across the surface of your cells like rafts on a liquid ocean.
That's the reality of it. And this led directly to Singer and Nicholson's famous 1972 fluid mosaic model.
Right, the fluid mosaic.
Yeah, the membrane is a 2-dimensional fluid where lipids and proteins drift around sideways, like tiles in a moving mosaic.
But, and here's where it gets really interesting, this creates a massive physical problem for the cell.
How so?
Well, if the hydrophobic effect builds this perfect, impenetrable lipid wall to keep water-loving molecules out, how does the cell actually eat?
Ah, right.
Like, glucose can't get through that lipid core. It's starting unless it has a back door.
That's where selective permeability comes back in. The membrane is highly discriminatory. Small, uncharged, lipid-soluble molecules, things like oxygen and carbon dioxide, they can diffuse directly through the lipid bilayer.
So they just passed right through.
Exactly. They move down their concentration gradient from an area of high concentration to low concentration. And it costs the cell 0 energy. That's simple diffusion.
Okay, but what about the bigger stuff? Or charged stuff?
For glucose or any charged ion, that hydrophobic core is an absolute barrier. They absolutely need to transport protein.
Wait, I always assumed that if there was a steep enough concentration gradient, stuff just naturally crossed over to balance out, like, water flowing down a steep hill. The physical pressure just forces it through.
That's a very common misconception, actually. A gradient is necessary, sure, but it is absolutely not sufficient on its own.
Really?
Yeah. Without a specific transport protein acting as a channel or a door, a charged ion is never getting through that hydrophobic core, no matter how steep the gradient is.
Oh, wow. So it's not just like a velvet rope at a nightclub letting anyone in if the crowd pushes hard enough on the street.
Not at all.
It's a highly specific coin-operated turnstile. The molecule has to be the exact size and shape of the quarter required to unlock the physical gear. Anything else, even a closely related molecule, just jams the slot.
That's a great analogy. And these turnstiles are floating in that fluid mosaic.
But wait, if the environment is completely fluid, couldn't the membrane just melt apart if you get a fever? Like if things get too hot?
So that's where cholesterol comes in.
Cholesterol?
Yeah, we often hear about cholesterol clogging arteries, but in the cell membrane, it acts as a bidirectional thermostat, a fluidity buffer.
A bidirectional thermostat. So if your body temperature gets too hot and the phospholipids start moving too fast, cholesterol acts like molecular glue.
Exactly. It restrains that movement so the cell doesn't get leaky.
But if things get too cold, the phospholipids want to pack tightly together and freeze into a rigid gel. So cholesterol acts like molecular antifreeze.
You got it. It wedges itself between those tails and forces them to stay apart. If cholesterol wasn't there acting as antifreeze, our cells could physically freeze solid on a cold day.
That's incredible.
And many organisms push this temperature control even further. It's a process called homeoviscous adaptation.
Homeoviscous adaptations, say that 3 times fast.
Right. But take cold water fish, for example. They physically alter the composition of their membranes. They incorporate more unsaturated fatty acids.
Okay, why unsaturated?
Well, these fatty acids have distinct kinks in their tails that prevent tight packing. So it ensures their cells stay fluid and functional even in freezing ocean temperatures.
Wow. Okay, so the membrane is fluid, the temperature is buffered, but we have to get back to that massive energy toll from the beginning of the deep dive, the heavy lifters.
Yes, the active transport pump.
Let's talk concrete examples.
The big one is the sodium-potassium ATPase pump.
Yeah.
Discovered by Jens Christian Skow in 1957, actually won a Nobel for it later.
And active transport means it moves substances against their natural gradient, right?
Exactly, which requires cellular energy in the form of ATP.
So for every single molecule of ATP it burns, it forces 3 sodium ions out of the cell and drags 2 potassium ions in.
Right. Both are moving against the direction they naturally want to flow.
It's basically bailing water out of a leaky boat while simultaneously pulling in a very specific type of fish. And the energy cost for this one task?
It's staggering. This single type of pump consumes roughly 1/5 to 1/4 of the human body's entire resting energy budget.
That is just, wait, really?
Yeah.
That means 1 out of every 4 breaths you take, 1 out of every 4 bites of food you eat isn't going to moving your muscles or thinking thoughts. It is going exclusively to this microscopic pump bailing out sodium.
It's wild to think about. And if you look at a neuron, up to 2/3 of its energy goes to this pump.
2/3.
Yeah, because maintaining those specific sodium and potassium gradients is the fundamental requirement for nerve cells to fire electrical signals. Without that pump constantly working, your nervous system instantly shuts down.
Well, another turnstile that just blows my mind is the aquaporin, because for a long time, scientists argued about how water actually got into cells, right?
They did, yeah. It diffuses a tiny bit to the lipids, but nowhere near fast enough to sustain life.
Right. And then in 1992, Peter Akori discovered this specific protein channel just for water, and he got a Nobel for that, too.
He did.
A single aquaporin channel conducts 1 billion water molecules a second. But the mechanism is insane to me. It lets 1 billion water molecules through, but it expertly excludes protons.
And that's harder than it sounds, because a proton is essentially a bare hydrogen ion, and in biological solutions, water and protons behave in very chemically similar ways.
So it's super easy to confuse them.
Exactly. For a channel to be open enough to let 1 billion water molecules rush through every second, but structurally precise enough to not let a single proton slip by and disrupt the internal pH of the cell, nature really engineered an absolute masterpiece of selective permeability.
These turnstiles are so incredibly precise. But what physically happens to your body when these specific gates are hijacked or broken? I mean, this is where microscopic biology slams right into human medicine.
Absolutely. And cholera is perhaps the most visceral example of this.
Cholera, right?
The cholera bacteria produces a toxin that doesn't actually destroy the cell membrane. Instead, it hijacks the turnstile.
Oh, wow.
Yeah, it permanently locks open a specific channel called the CFTR chloride channel in your intestinal cells.
So the coin is basically glued into the slot, the turnstile is spinning freely, and chloride ions just absolutely flood out of the cells and into the intestine.
Exactly. And because of osmosis, water chases the solute. Water rushes out of the intestinal cells to follow the chloride.
And that massive, uncontrolled fluid loss is the watery diarrhea that makes cholera so deadly.
Right. The physical wall is entirely intact, but the transport controls are completely subverted.
That is terrifying.
And what's tragic is that this exact same channel, the CFTR channel, is the culprit in cystic fibrosis.
Wait, really? The same channel?
The exact same one. But instead of being locked open by a toxin, it's broken by a genetic mutation, usually delta F508. The channel simply doesn't work. It won't open.
And because the chloride gets trapped inside the cell, the water stays inside the cell with it.
Yes.
So that means the mucus on the outside of the cell doesn't get hydrated. It turns into this thick, sticky glue that clogs the lungs and invites persistent infection.
Exactly.
Yeah.
A lethal multi-organ disease, all because one microscopic door won't open. And, clinical manipulations of these membranes are everywhere. A local anesthetic like lidocaine works by physically blocking voltage-gated sodium channels in your nerve cell membranes.
So the sodium can't rush in, the electrical potential can't fire, and the pain signal never even reaches your brain.
Precisely. Or take antibiotics. Penicillin destroys bacterial cell walls.
Oh, and we have to clarify a major mechanism here just to bust a common misconception, because people constantly confuse cell walls and cell membranes.
They do all the time.
Plant cells, fungi, and bacteria have a rigid structural cell wall on the outside of their flexible membrane. Animal cells, our cells, do not have a cell wall. We only have the flexible membrane.
Which makes our cells much more flexible in shape, sure, but incredibly vulnerable to osmotic pressure.
Exactly. Think of an animal cell like a water balloon. If you put it in pure water, osmosis forces water inside to balance out the sleuths. Because there is no rigid cell wall to push back and contain that pressure, the animal cell will swell up and literally pop.
Which is exactly why the IV bag they hook you up to at the hospital is never plain water.
Right. It's an isotonic saline solution. It's precisely formulated to match the exact solute concentration of your blood so your red blood cells don't swell and burst.
Exactly. And returning to penicillin, when the antibiotic destroys the bacterial cell wall, it leaves the bacteria's underlying flexible membrane totally unsupported.
So it just pops.
Yep. The internal pressure of the bacteria causes the membrane to rupture, killing the cell entirely.
Okay, so we've mapped this transition from a static cracker sandwich to a fluid mosaic of drifting turnstiles. And, it's tempting to treat the 1972 fluid mosaic model as the finish line. Like, we figured it out, put it in the textbooks, case closed.
But that would be a mistake.
Yeah.
The physical reality of the membrane is far more complicated, and the scientific debate is very much alive today.
Right, because science didn't stop in 1972.
Not at all. One major ongoing area of research involves lipid rafts, which were proposed in 1997.
Lipid rafts. Okay, what are those?
The idea is that the membrane isn't a totally random uniform sea. There are highly structured zones, these small patches enriched with cholesterol and specific lipids, acting like floating islands.
So they organize signaling proteins into functional clusters so they can work together more efficiently.
Exactly. But they are highly controversial.
Why?
Because they are too small and short-lived to clearly see with standard microscopy. So you have some scientists who swear they're essential biological platforms, and others who think they might just be physical artifacts created by the chemicals we use to prepare membranes for study.
So the jury's still out.
Very much so. And the structural challenges go even deeper with Akihiro Kusumi's picket fence model.
Picket fence model? I haven't heard of that one.
Well, with the advent of high-speed single-molecule tracking, researchers can finally watch individual proteins move in real time. And what they saw directly challenges the fluid, wide-open ocean concept of the fluid mosaic model.
Wait, really? So they aren't just floating freely anywhere they want?
No, it's not a wide-open ocean at all. It's more like a series of microscopic sheet pens.
Sheep pens?
Yeah, the cell has an underlying cytoskeleton, a network of actin filaments just beneath the surface. This acts like a physical picket fence, corralling the membrane proteins into tiny compartments.
Oh, wow. So they bounce around inside one pen for a bit, then occasionally hop the fence into the next corral.
Exactly. They're highly constrained.
So if the textbook sandwich model was wrong for 35 years, and now the fluid mosaic is being heavily revised with picket fences, what does that tell you as a learner about scientific truth?
If we connect this to the bigger picture, the Davson-Danieli failure is highly instructive. It reminds us that a consensus model can be built carefully on the absolute best data available at the time. It can make apparently confirmed predictions, and it can still be fundamentally wrong about the underlying structure.
Yeah.
Science is about constantly refining our models as our tools, like freeze-fracture microscopy or single-molecule tracking get better. We are perpetually moving toward a higher resolution of reality.
That is perfectly said. I mean, we started this deep dive looking at what we assumed was just a passive biological wrapper. But this microscopic border is a highly structured, corralled mosaic. It is a dynamic wall manned by billions of molecular turnstiles that consume a massive percentage of your daily energy just to keep you alive.
It truly is the definitive gatekeeper.
It is. And I want to leave you, the listener, with a final thought to mull over as you go about your day. We established early on that the foundational architecture of the cell membrane, the lipid bilayer, is chemically inevitable. It self-assembles perfectly just by mixing lipids and water, driven entirely by thermodynamics and the hydrophobic effect without using a single drop of cellular energy.
Right.
So, if the very boundary that separates the living interior of a cell from the dead, chaotic outside world builds itself automatically through blind physics, at what exact microscopic point in that self-assembling boundary does chemistry actually become biology?
That is the big question, isn't it?
It really is. Keep questioning the world around you. We'll catch you on the next deep dive.